Hi everyone, I am Nur Shazwani and my metric number is 202345. So in this video, I'm going to do two experiments which are strip plate method experiment and Gram staining experiment. So these are the materials that is needed in strip plate method experiment, which are nutrient plate agar. I use tap water as a broth. I use candle as a Bunsen burner, permanent marker, and inoculation loop. So first of all, the principle of this experiment is to isolate the discrete colonies from mixed culture. The concept is like dilution of the culture, which is from the high concentration region of bacteria to the lower concentration region of bacteria, and at the end of the experiment, you will obtain the single colonies. So first of all, using permanent marker, label the bottom of the dish by drawing two bisecting lines to divide the plate into 4 equal parts. And label each part from number 1 to number 4. Next, sterilize the loop by flaming it and let it cool for a while before dip it into bacterial culture. Place a loopful of culture on the agar surface in quadrant 1. Quickly spread the bacteria throughout the quadrant. Proceed to the next step. Reflame and cool the loop and turn the petri dish into 19 degrees. And then touch the loop into the quadrant 1 and drag it across the quadrant 2. Repeat the same step by reflame and cool the loop and turn the petri dish into 90 degrees. Then touch the loop on the second quadrant and drag it into the third quadrant. Again, without reflaming the loop, turn the dish into 90 degrees and place the loop on the third quadrant and drag the loop to the fourth quadrant. As the expected result for this experiment is, you will get the single colony of bacteria on the fourth quadrant. Such as the example, I use Serratia marcescens, which is actually the curry powder, to construct the expected result because Serratia marcescens has distinctive color, which is easier to be seen. As you can see, there were heavy confluent growth of bacteria on first and second quadrant, and the light growth were shown on fourth quadrant. From it, you can obtain the single colony. So now let's move to the next experiment, which is Gram stain. These are the materials needed for the experiment, which is I use tap water as a decolorized agent and also tap water. I use 3 types of color as reagent, which is iodine, crystal violet, and safranin. I use candle as a Bunsen burner, inoculating loop, permanent marker, syringe as a dropper, slide, tissue as a bibulous paper, staining tray, and basin. So the principle of this experiment is, it is used as a tool for classification and differentiation of microorganisms. From this experiment, we also can obtain the 2 major groups of bacteria, which are gram-positive and gram-negative. There are at least 4 chemical reagents in this experiment, which are primary stain, which is crystal violet. It is used to impart its color to all cells, and the color of cell will turn purple. The second reagent is gram iodine, which is mordant. It's used to intensify color of primary stain. so the cell will become black purple. For the third reagent is decolorizing agent, which is alcohol. Its function depends on two factors, which are concentration of lipid and the thickness of the peptidoglycan. So, alcohol will remove the color on the gram-negative bacteria and leave the gram-positive bacteria cell remain purple. Because positive gram-bacteria, it has thicker peptidoglycan layer. For the fourth chemical reagent is safranin, which is contostain. It will stain the decolorized cell with pink color. So, in this experiment, the negative gram-bacteria cell will absorb the pink color. Before we proceed with gram staining experiment, we have to do preparation of bacterial smears, which is for the microscopic visualization of bacteria. So, in order to do bacterial smear, first of all, label the bottom side of the slide by circle it at the center of the slide. Next, by using aseptic technique, place a loopful of culture on the other side of the slide and spread the culture by doing the circular motion inside the circle. Allow the smear to air dry for a few minutes and then do the heat fix by quickly pass the smear over the flame 2 to 3 times. By doing the correct technique, you will get the good smear, which is, when dried, it will appear as a thin whitish layer on the slide. Move to the gram staining experiment. By placing the slide on the staining tray, gently flood the smears with the first reagent, which is crystal violet. And let it stand for 1 minute. After 1 minute, gently wash off the stain with tap water. Next, gently flood smears with the grams iodine mordant and let stand for 1 minute. After 1 minute, gently wash off the grams iodine with tap water. And for the next step is decolorize the bacterial cell with 95% alcohol. This is an important step and must do correctly to prevent over-decolorized. Add the reagent drop by drop until it's showing only blue tinge. Over-decolorized will cause the safranin stain cell will appear to be darker in the color. Thus, it could be mistaken for a light purple gram-positive stain cell. Then, gently wash off the 95% alcohol with tap water and counterstain the decolorized cells with safranin for 45 seconds. After 45 seconds, gently wash off the safranin with tap water and blot dry with bubbleless paper. The expected result for the experiment is, when you observe the stained bacteria using microscope, you will get to observe the morphology and the arrangement of the cells and also the different color, where gram-positive cells are purple and gram-negative cells are pink. Okay, that's all from me. Thank you.